It's not strong enough. Okay, so that's just about osmotic, but the other tests that can be done are listed, but you may not bother yourself about that. Okay, so this is how spherocytes looks like. They are circular, they are not biconcave disc, and the area of central pallor is almost absent. Okay, and this is how elliptocyte, you can see the elliptoid cells predominant. Some people have inherited this from their parents, so you can see the elliptoid cells, red cells. So this is hereditary elliptocytosis, and this is, so they are also mutated of the alpha and beta spectrin. Red cell is elliptical, and this is also another diagram showing A and A, they are elliptoid cells, hereditary. So if you see many of these red cells on the field, then you can say this person has probably hereditary elliptocytosis. So what test will you now do? Osmotic fragility test. Osmotic fragility test, okay? This hereditary pyrrhopoikilocytosis is also, you can see the Southeast Asia ovalocytosis. This is another type. This is stomatocytosis. You can see the slit-like mouth in the red cell. It's like a mouth, okay? Showing like a mouth. This is hereditary stomatocytosis, okay? So this is about osmotic fragility, which I've described osmotic fragility test, okay? So this is the concentration. You can see the test tubes in various concentration. So the darker the darker the tube, the more the hemolysis. You can see the sodium chloride concentration, and then how it is diagnosed. Okay, so this is how hereditary osmotic fragility test is done, and these are the other tests you can do to confirm hereditary. So this is hereditary. So we talked about the sodium deoxyribonucleic page. Okay, these are the proteins, red cell membrane. So this, if you want to extract, if you want to, this is what is called sodium deoxyribonucleic sulfate polyacrylamide gel electrophoresis. Page is polyacrylamide gel electrophoresis. Okay, the salt that is used is like you want to use a salt to extract out the membrane, the protein in the membrane, and run them under. And have you watched hemoglobin electrophoresis, how it's done? Hemoglobin electrophoresis, have you watched how it is done? No. Okay, but so this is a form of electrophoresis called polyacrylamide gel. They use a gel to separate out the proteins of the red cell, and then use a stain called Kumasi blue stain to stain it, so that you can see whether alpha spectrin is present, beta spectrin is present, and carrin is present. So you have a gel, okay? You extract the membrane protein with a salt. This is sodium deoxide sulfate salt. That's why it's called SDS. You extract it out from the solution, and then you now run the protein on electrophoresis. So the protein will move according to their molecular weight and charge on the electrophoretic tank. Okay, after that, you now stain. But for carbohydrates, what is used for staining is periodic acid skip stain, and that's why biacophorin A, glyphosate can be, so it's done separately. But for proteins, these are, so if you now have all the proteins lined up, and see which one is present, which one is missing. So you can now say, oh, the hereditary spherocytosis, it is because the spectrin alpha spectrin and beta spectrin are missing after your electrophoresis, and then you can confirm. I hope that is clear. It's not. It's not. So we'll talk about enzymopathy, and this will be more boring for us, except those that are good in biochemistry. Okay? Because I did a first degree in biochemistry, so don't be surprised that I'll be talking a lot about biochemistry, and it's still very helpful. So it is the deficiency of red cell enzymes. It is usually hereditary. So these are the content of the red cell. It is just red cell enzymes and hemoglobin. Okay, so we'll now talk about the content now. Red cell enzymes. Okay, and then don't forget there is no nucleus. It's just the cytoplasm that has the red cell enzymes, and then that's hemoglobin that is occupying all the space inside the red cell. Okay, almost all the space. Okay, so some people have inherited deficiencies of some of these red cell enzymes, and that is what enzymopathy is all about. Can you give me an example of a red cell enzyme that is common? G6PD, glucose 6-phosphate dehydrogenase deficiency. Okay, it's a red cell enzyme. We know about G6PD deficiency. Okay, we've heard about it. Okay, so it may be divided into 3 groups, and these are the disorders that affect the ability to deliver a rapid response to oxidant stress generated in red cell reactions, mainly deficiency in the pentose phosphate pathway and then the glutathione cycle. You still remember our pentose phosphate pathway? Do we remember pentose phosphate? Do we remember? But I'm sure we remember the glycolytic pathway. We remember. Who will tell me about the glycolytic pathway from glucose to glucose 6-phosphate to don't worry, by the end of the lecture. So the importance of these enzymes is that they generated that of the pentose phosphate generated NADPH, and that of the glycolytic pathway generates ATP. Don't forget you need ATP for your sodium, potassium, ATPase, and then the calcium. That's where it comes from. So if you have deficiency of all these red cell glycolytic pathway enzyme, then will you have plenty of ATP? No, there will be no plenty of ATP. And if you don't have plenty of ATP, there will be reduced active transport, because there is no ATP to power the sodium potassium ATPase, and then the sodium will accumulate in the cell. Instead of pumping 3 sodium out, you now be having 1 sodium out, 2 sodium at best, because there is no ATP. Then potassium will now accumulate outside instead of bringing potassium, and so the implication is that the cell will swell and then burst because of deficiency. What about the NADPH that is generated from pentose phosphate pathway? How will it affect survival? We don't know, but don't worry. So there is miscellaneous enzymes deficiency where mechanism of hemolysis is not too clear. Okay, so most of the red cell enzymes participate in reactions which lead to generation of reducing power in form of NADH and NADPH. Okay, and the production of energy in forms of ATP, adenosine triphosphate. So the reaction system include the pentose phosphate pathway and the glutathione metabolism, and then the glycolytic pathway. These are the 3 major pathways that are involved in the generation of all this reducing power in form of nucleotide adenine dehydrogenase phosphate deficiency, that's the NADPH, or nucleotide adenine dehydrogenase NADH, or adenine triphosphate, which is your ATP, are generated from the PPP pathway, pentose phosphate pathway, and the glutathione pathway, and then the glycolytic pathway that generates ATP. Okay, so this is your, and then the link. You can see the glycolytic is also known as MD-Meyer, if you remember. MD-Meyer, your glucose will be converted to glucose 6-phosphate, and glucose 6-phosphate will be converted to glycerol 3-phosphate. Glycerol 3-phosphate will be converted to eventually phosphoenol pyruvate and lactate. But your glucose 6-phosphate that is generated in the pathway, you know, is fed into pentose phosphate pathway to form a 6-phosphogluconate, and 6-phosphogluconate is converted to ribulose 5-phosphate, and in the process 2-NADPH is generated. Okay, 2-NADPH is generated. Look at the third glutathione, how it's involved. The NADPH is needed to reduce oxidized glutathione. This is an oxidized glutathione into reduced glutathione. So why do we have to convert oxidized glutathione to reduce glutathione? You need the reduced glutathione, which is GSH, to do what? To break down oxidant radicals in the cell, because the cells generate hydrogen peroxide that are toxic to the cells. So you need GSH, which is a reduced glutathione, to break it down to water so that they become less toxic. And then the superoxide is also generated, and that's what is responsible for aging. And that's why some people take antioxidants. You know, if you take antioxidants, you are going to aid the breakdown of byproducts of the cell, which is hydrogen peroxide and superoxide, and break them down into less toxic. And that is what is causing our skin to wrinkle, our eyes to be baggy like my own. As you age, you generate a lot of oxidants, the oxidants in your cells, and the oxidants destroys the cells, except you have this mechanism in place. So okay, so that's, so the maintenance of biconcave disc of red cell shape, which is complete with the survival for 120 days, require expenditure of metabolic energy. We've agreed on that. ATP, NADH, NADPH. Okay, so without it, if you cannot generate it, then the 120 days lifespan will be shortened. Okay, it will be shortened. Okay, so the system that maintains the shape and require the energy for the maintenance are the sodium potassium ATPase, which ensures high potassium and low potassium levels intracellularly and vice versa extracellularly. Phosphorylation of membrane cytoskeleton proteins, which ensure biconcave disc. So this reduces the main function of red cell, which is the binding, transport, delivery of oxygen to tissues, do not require energy expenditure. The reducing power is, however, essential for other red cell function. This is because the oxygen carrying protein of the red cell, you know, hemoglobin, can bind, transport and deliver oxygen to tissue only if it exists in its reduced form, and it requires the NADPH to convert it from the oxidized form into reduced form. The oxidized form of hemoglobin has iron in 3 pluses. The reduced form is iron in 2 pluses. So you need NADPH. So if the hemoglobin you are talking about will not be able to work if you don't have adequate NADPH. Okay, so you need NADPH to convert oxidized hemoglobin into reduced hemoglobin. So maintaining the iron of the hemoglobin in the divalent form and also maintaining the sulfhydryl groups of red cell enzymes, membrane proteins and hemoglobin in the active reduced form. Okay. The extraction of energy from substrate and generation of reducing power, okay, requires participation of a large number of enzymes. Okay. So the red cell enzymes are not replenishable as the cell is anucleated. Okay, it has no nucleus. The enzymes are synthesized from the nucleated marrow cells and to some extent by reticulocytes. Okay, so the main substrate for red cell energy production is glucose, while adenosine, inosine, mannose and other can also be used to a little extent. Okay, so you can see I want to look at this diagram again. Glucose 6-phosphate is this is the PPP, pentose phosphate. You have the 6-phosphogluconate, and in the process you generate NADPH. NADPH is needed to reduce methemoglobin, which is hemoglobin with 3 pluses of iron, to and this is the active form of that's the reduced hemoglobin. So you can see why. So you use then the NADPH is also needed for reduced glutathione, which is GSSD, to convert it to only oxidized glutathione is GSSD to reduce glutathione. The reduced glutathione, this is hydrogen peroxide, which is toxic to the cell. You need reduced glutathione to break it down to water, and then if you don't do that, the catalyst, will cause the conversion of reduced hemoglobin to oxidized hemoglobin, and then it becomes less effective. So you can see the link between the glycolytic pathway, the PPP. So the pathways are just three: the PPP, the glutathione cycle, and the glycolytic pathway. And this is the PPP. You have the oxidative and the non-oxidative. Glucose 6-phosphate coming from the glycolytic pathway is converted to 6-phosphogluconate, and then one NADPH is generated. 6-phosphogluconate is converted to 6-phosphogluconate, and then 6-phosphogluconate is converted to ribulose 5-phosphate, and another NADPH is generated. So you have two NADPH, okay, from that pathway, and then the NADPH will be used to convert oxidized hemoglobin to reduced hemoglobin, which is hemoglobin with iron of 2 pluses. The NADPH will be used to convert oxidized glutathione to reduced glutathione. The reduced glutathione will be used to convert hydrogen peroxide to water. So if there is no NADPH, the hydrogen peroxide will be converting reduced hemoglobin to oxidized hemoglobin, and it will be useless. I hope we are on the same page. So this is the non-oxidative pathway, and then you are going to generate glycerol 3-phosphate and fructose 6-phosphate. So what is, okay, there is an enzyme that is needed, which I forgot to mention. This is the glucose 6-phosphate dehydrogenase. So if you have deficiency of glucose 6-phosphate dehydrogenase, will this pathway move forward? Will you have adequate NADPH? Can you see the link? So some people have inherited glucose 6-phosphate dehydrogenase from deficiency from their parents. So if you have inherited glucose 6-phosphate dehydrogenase deficiency from your parents, then you will have challenge with production of NADPH, because even if you cannot convert glucose 6-phosphate to 6-phosphogluconate, you cannot even get to this point. Okay, you can't get to this point, so there will be complete lack of NADPH. If you lack NADPH, you know the implication. You cannot convert oxidized glutathione to reduced glutathione, and if you don't have reduced glutathione, you cannot break down hydrogen peroxide. Apart from that, if you lack NADPH, you cannot convert oxidized hemoglobin into reduced hemoglobin. Oxidized hemoglobin has 3 pluses of iron, which is non-functional. Reduced hemoglobin is the one that can bind oxygen, carry oxygen to the various sites where it is needed, and then deliver oxygen. If you deliver oxygen, then the aerobic cycle will be used. In anaerobic cycle, how many ATP is generated? Eh? Two ATP is generated. So, but with aerobic cycle, you'll be able to generate your NADPH along with the ATP. So, in GC-PD deficiency, the commonest and the most studied enzymopathy. You know, it's over 200 million cases worldwide. It's so common. Okay, GC-PD deficient. Okay, so it is the only source. It's the only source of NADPH in the red cell. It's the only source. So if you are deficient in GC-PD, so the GC-PD gene is located on the X chromosome, and so it affects predominantly females. Males affect predominantly males. Did you switch off the AC? Okay, no problem. So it affects predominantly males, and that's why GC-PD affects predominantly males. So there are more than 400 variants of the enzymes. Okay, because you have isoenzymes and so on and so forth. And there are two main knowns. The A variant, which is the African type, and the B, which is the Mediterranean type of GC-PD. Okay, so the A type and the B type. You know the implication of being GC-PD deficient from what you have said. So there are some drugs that will generate, if you take the drugs, it's going to generate plenty of oxidants, like the hydrogen peroxide, superoxide. So if you give such drugs to somebody that is GC-PD deficient, you have plenty of hydrogen peroxide, there is no NADPH to break it down, and then there will be hemolysis. Okay, apart from that, there will be no NADPH to convert the oxidized hemoglobin to reduce hemoglobin, and then there will be anemia, because the hemoglobin cannot carry oxygen. So like drugs like anti-malaria, primaquine and pamaquine can trigger sulfonamides, sulfanilamide, sulfapyridine, sulfadimidine, sulfacetamide can trigger hemolysis in GC-PD deficient individual. Okay, miscellaneous drugs like nitrofurantoin, nalidixic acid, beta-adaptor, acetyl salicylate. Acetyl salicylate is what? Otherwise known as aspirin. And then methyl blue, nilidazole, naphthalene, mothballs. You know, in those days, some grandmother would put mothballs in the shawl of a baby that may be GC-PD deficient, and then the person will start hemolyzing, you know. So those are the drugs that can trigger hemolysis in GCSPD deficient individual. So infections can also cause hemolysis. Gram positive, gram negative infections can cause hemolysis. Diabetic ketoacidosis, fava beans, fava beans. You are going to do GCSPD deficiency in pediatrics when you pass from this level, and you will be able to appreciate the pathogenesis. If you go back to this slide, you'll be able to, because they may not even know what you are being taught now. The pathogenesis, the linkage with generation of NADPH, the linkage with pentose phosphate pathway, the linkage with the glutathione cycle. Is when you understand that you'll be able to appreciate the pathogenesis of, so all these things that can trigger oxidant stress in red cells if patient is exposed, drugs, infection, and then fava beans. Fava beans, I've never seen one before, but it's a type of beans that can trigger. So GCSP deficient red cell are more susceptible to than normal red cell to oxidative damage. Okay, so this is because NADPH produced by GCSPD is required. We explained this for generation of glutathione, reduced glutathione, and in turn detoxifies hydrogen peroxide by glutathione peroxidase. The oxidase can explain this. So this is the same diagram explaining how GCSPD deficiency, if it's deficient, you lack NADPH. If you lack NADPH, you will not be able to reduce oxidized glutathione, to convert oxidized glutathione to reduce glutathione. You need a glutathione reductase, and if you don't have reduced glutathione, you cannot break down the oxidant to water, and then the cells become toxic. The oxidant will break down the red cell. So this is a glutathione cycle, and the same diagram, NADPH is needed to convert oxidized glutathione to reduced glutathione. Reduced glutathione is needed to convert hydrogen peroxide to water. So if you have accumulation of hydrogen peroxide, it's going to be toxic and break down the red cell membrane despite its strength, despite its durability, despite all the vertical interactions, despite all the horizontal interactions, despite all the lipid bilayer, which are aminophospholipid and choline phospholipid. The vertical interactions, which are spectrin combined with it can be band 3, acarine, spectrin, it can be glycophorin, protein 4.2, and spectrin. And the horizontal interactions are the alpha and beta spectrin that can form a tetramer. So despite all this, if you have too much of hydrogen peroxide, it's going to break it down. If you have too much of superoxide, it's going to break it down. Okay? So this is another diagram showing, so there's a superoxide dismutase. This is another oxidant that can cause, apart from hydrogen peroxide, this is where hydrogen peroxide is formed. You have oxygen being converted to superoxide, which is 0,2 with hyphen on top, and then it can be converted to hydrogen peroxide. The superoxide can be converted to oxygen. Okay, SOD is superoxide dismutase. Superoxide dismutase. So you need NADPH to convert the oxygen to superoxide and then you have NADP, and then the superoxide is converted to hydrogen peroxide. The superoxide can cause oxidative damage and organ dysfunction. Okay, so you need NADPH. So this is another diagram showing the linkage between the glycolytic pathway, pentose phosphate pathway, and then the glutathione cycle. Okay, so this is PD deficiency is usually asymptomatic, but acute hemolytic anemia occurs when the subject is exposed to oxidant stress, like I've mentioned, drugs like primaquine, pharmaquine, sulfur drugs like sulfatimidine, sulfacetamide, and sulfadilidine, and so on and so forth. Okay. Then fava beans, I mentioned that, or infection, diabetic ketoacidosis. The severity of hemolysis can vary from causing mild anemia and jaundice. So when there is, what's the link between hemolysis and jaundice? The link between hemolysis and jaundice. Okay. Hemoglobin is released. Hemoglobin is broken down to hemoglobin and iron. Then heme is broken down to biliverdin by what enzyme? Heme oxygenase. And biliverdin is converted to by what enzyme? By what enzyme? Biliverdin reductase. Biliverdin reductase. And so you have accumulation of what? Accumulation of bilirubin. You have accumulation of bilirubin, and that is what is responsible for jaundice. Okay, we are going to talk about hemoglobinopathy, and then we'll talk more about that. Okay. So GC-PD deficiency is a common cause of neonatal jaundice in Nigeria. It rarely causes lifelong chronic non-spherocytic hemolytic anemia. So it's an abrupt onset. This is so a patient presents with hemoglobinuria. What is the color of urine in hemoglobinuria? It's usually coca-colored urine, because there's plenty of hemoglobin that is released, and then some of them are passed into the kidney like that, causing excretion of hemoglobin whole without being broken down to the hemoglobin, and then it will cause Coca-Cola colored urine in the child. So jaundice is also possible, renal failure, because the hemoglobin can clog the kidney nephros and then destroy the kidney, cause renal failure, and then reticulocytosis in day 5 to 8, because the body will be trying to compensate for the reduction. Okay. So there is during crisis, blood film may show contracted or fragmented cells called bite cells or blister cells. You can be asked, where is bite cell or blister cell seen? Is GC-PD. Is a common practical question and a common MCQ question. Anywhere you see bite cell or blister cells, so it's GC-PD deficiency. Okay. So this is glutathione. Glutathione, you have the reduced glutathione, which is GSH. You have the oxidized glutathione, which is GSSG. Okay, and we've seen the linkage between the glutathione that is reduced and the oxidized glutathione. So this is glutamate. Glutathione is made of 3 amino acids: glutamate, cysteine, and glycine. Okay, glutamate, cysteine, and glycine. Okay, so that is the diagram of the glutathione. That is the diagram of the glutathione, and this is how we've explained this before now. How superoxide is converted to hydrogen peroxide by superoxide dismutase. Okay, and the hydrogen peroxide must be broken down to water by catalase. Okay, and for that to happen, you need a reduced glutathione to be converted to oxidized glutathione, and to convert reduced glutathione to oxidized glutathione require NADP, NADPH. Okay, so you need a reduced glutathione reductase is needed to convert NADPH into NADP, and glutathione peroxidase is needed to convert the hydrogen peroxide into water. Okay, so superoxide dismutase converts this superoxide into. So there was a time we carried out a research on sickle cell, and we assayed this enzyme superoxide dismutase in sickle cell patients and in AA individual. Do you think it will be high in sickle cell or low compared to AA based on what you have discussed? The SOD, superoxide dismutase, will it be high or low? It will be high because it's contributing to, but if it's high, it will be converting into hydrogen, or should it be low? It will be low, or whatever. It may be, I think we got high, I'm not sure. It will be high. You can Google it. We did a study where we assayed superoxide dismutase in sickle cell and then compared it with AA. So we've discussed the oxygen-free radicals, superoxide and hydrogen. These oxidant stress are produced in number of reactions, such as methemoglobin formation, oxygen burst of macrophages, and they must be converted to less toxic form, which is water. And then you need NADPH to do that. You need reduced glutathione to convert it, and you need NADPH to get the reduced glutathione. And the NADPH is from glucose 6-phosphate dehydrogenase. So this is the other. So the glycolytic enzyme deficiency, they are usually autosomal recessive except the phosphoglycerol-kinase, which is X-linked. Apart from that, pyruvate kinase deficiency can convert pyruvate to lactate. It's also the most common after G6PD. Pyruvate kinase deficiency is the most common after G6PD. So this is the glycolytic pathway, and you can see the phosphoglycerol-kinase where ATP is generated, which is the S-linked, and you can see the pyruvate kinase that converts phosphoenol pyruvate to pyruvate. Okay, so this is the glycolytic enzyme, and then these are the how they present. If there is no pyruvate kinase, if there is no exokinase, if there is no glucokinase, you will not be able to generate ATP, and if you cannot generate ATP, then you cannot use it for the sodium potassium ATPase. Okay, so pyruvate kinase deficiency also have two form of enzymes, the liver and the muscle forms, and diagnosis is autoimmunosis of red cell not corrected by glucose, or like hereditary spherocytosis. Treatment is to use folic acid, avoid iron arbitration, and splenectomy. So the other enzymes, apart from pyruvate kinase deficiency, all other enzymes, enzymopathy involving glycolytic are very rare. All other enzymes could also be deficient, but they are not common. Glucokinase, exokinase, and phosphokinase, and all the rest are also very rare. The clinical and hematological picture in all these enzyme deficiency is that of a lifelong hemolytic anemia. Okay, so I just want to, this is another enzyme that can be deficient and cause hemolysis, pyrimidine 5 nucleotidase. In contrast to other enzymopathy, LCM morphology is relatively characteristic in that it exhibits prominent basophilic stiffening. Okay, so pyrimidine 5 nucleotidase deficiency is another enzyme apart from the glycolytic enzyme deficiency and the G6PD deficiency. So they will all cause. You want to rest before we start hemoglobinopathy. Next week, you have another lecturer. That means you will not have it until after your lectures. And there is thalassemia coming up next week. You need a good knowledge of hemoglobin